Anyway, it's a pleasure to welcome Reza Dana. I've known Reza for I don't know how many years now, but quite a few, back when we both had more hair and less grey. And it's just a treat to have him here. We've met up and talked science in many beautiful parts of the world, I'm happy to say. Bermuda comes to mind, and Berlin and many other cool places. It's for you trainees, if you really can't get into the science, stick with it because the travel is pretty good. So Reza has been at Harvard for, what's it, 20 years now, Reza? 20 years, my God. He's now professor of ophthalmology. I'm having to read this because he has so many titles. He is vice chairman of the Harvard Department of Ophthalmology. He's associate chief of ophthalmology for academic programs at Mass Eye and Ear. He is the co-director of research, senior scientist at the Scapin's Eye Institute, and the director of the cornea and refractive surgery at Mass Eye and Ear. But we didn't bring him here for all that. We brought him here because he does really good immunology in the cornea. He's certainly been one of the leaders, pioneers in the field in this area, both in T-cell immunology and responses in the cornea and in lymphangiogenesis in particular, most recently. So Reza, pleasure to have you. Thank you for coming.
Thank you so much, Eric. That was very kind of you. It's great to be here among friends. Eric, Jonathan, it's a pleasure to see what you've done to the department. It's been a relatively short time, and you've accomplished so much. Really, every time that I come back, I'm amazed how much you've done. It's very nice and impressive to have this group come together, a truly multidisciplinary group to talk about vision and the eye world. So understanding the highly heterogeneous group here this afternoon, I wanted to give you sort of a high-level view of some of the basic pathological phenomena on the ocular surface, and then hone in on some pathophysiologic mechanisms, and then talk about relevance. I had put together a talk as of this morning that was largely focused on translational aspects of eye research, and then in talking to Eric this morning, he told me, well, actually the audience is entirely scientists and no clinicians except for one or two people. So I've taken some of the translational and clinical aspects off, and we'll focus more on the science. To begin with, in terms of disclosures, I do serve as a consultant for a number of biotech and pharmaceutical companies, and I'm also listed as an inventor on a few patent applications. So to put this area of corneocular surface in perspective, it's really important to emphasize how relevant it is from a public health standpoint. I like to begin this way always in Boston because our department historically has been so retina focused and probably not so here. But it is good to remind ourselves that after cataract, corneal disease is the leading cause of non-refractive blindness worldwide. And there is nothing more devastating to the cornea as a corneal angiogenesis, almost as an end product of many severe forms of inflammatory corneal disease. Corneal angiogenesis is pervasive. It's a consequence of infection, trauma, immune-mediated insults, and also, as some of you may know, it's a very significant risk factor in terms of the survival of corneal transplants performed for the corneal scarring and angiogenesis. So if you have a cornea that is this way, if you do a corneal transplant, oftentimes it fails because, as you'll see, the blood vessels are important conduits of inflammatory cells and alloreactive or graft-specific T cells. Now, the cornea is normally obviously avascular. It's one of the principal and interesting features of the cornea, except for the cartilage. It's the only other tissue that does not have any blood or lymphatic vessels. It's called angiogenic privilege. And this angiogenic privilege, as you'll see, is very tightly regulated, because once this is abrogated, then the ingrowth of blood vessels and lymphatics can significantly alter the immunohomeostasis in the tissue. So corneal blood vessels can, again, occur as a result of many different types of insults. On the left here, you can see Those of you who can't see, not that cluster over there. You can see the growth of blood vessels in a case of severe atopic keratoconjunctivitis. And on the right, we have a case of an alkali burn. This can happen chronically, and on the right, this can happen very quickly over a matter of 4 to 6 weeks. You can get very robust ingrowth of blood vessels. Now, there is a very interesting relationship between blood vessels and inflammation. Inflammation can induce blood vessels, and as you'll see, blood vessels and some other phenomena that it can potentiate can amplify inflammation and immunity. So there's a very wide array, many dozens of cytokines and immunoreactive factors that can induce angiogenesis. These range from cytokines such as interleukin-1 and TNF-alpha, IL-6 that we heard about earlier in the context of Th17 and PMN17s this morning, adhesion factors and proteases, all of which can either promote proliferation of vascular endothelial cells or lead to changes in the matrix, which will facilitate the migration of these proliferating endothelial cells prior to tubule formation. And virtually all of the inflammatory chemokines also can directly affect angiogenesis, except for a few here that lack the ELR motif, such as the ones that are driven by interferon gamma, like the CXCR3 ligands. And then, as I mentioned, angiogenesis can amplify inflammation and immunity. And the chief reason for that is that the principal source of inflammatory cells that go to cause infiltration of tissues, whether it's a corneal infiltrate or inflammation anywhere in your body, is the intravascular compartment. And the principal direct, if you will, factor that is proportional to the amount of inflammation that you can get is the surface area of the vasculature in contact with the matrix in which the inflammatory chemokines are being expressed. That would be giving these directional signaling to these cells that are going through the endothelium, what we call extravasation of these leukocytes. So angiogenesis and inflammation work together to amplify immunity and inflammatory responses. Now, one of the interesting things that we became interested in almost now 2 decades ago is the fact that if you induce angiogenesis, and angiogenesis is showed in these slides as green, we're standing here for CD31 or PCAM, is that if you induce angiogenesis such as placement of a corneal suture as shown here, within a number of days you can see the sprouting of lymphatic vessels shown here in orange due to their staining with live 1 antibody. Now, lymphatic vessels almost always accompany the ingrowth of blood vessels. And as you'll see later on, the converse is not true. You can have selective ingrowth of lymphatics without ingrowth of blood vessels under certain conditions. Now, what is the relevance of the ingrowth of these lymphatics, these orange vessels as shown here? And by the way, this panel on the right, here's the normal limbus. This is normally blood vessel and lymphatic free. And in response to inflammation, you can see this profound ingrowth of blood and lymphatic vessels. Now, the relevance of this, and I want you to keep this, this cartoon in mind as we go through this talk, is that virtually all forms of antigen-specific immunity involve coming in contact of antigen-presenting cells with either foreign antigens, whether they're microbial antigens or autoantigens, altered autoantigens or transplant antigens, what we call alloantigens. And then the migration of these antigen-presenting cells, APC, through lymphatics to the lymphoid tissue. The lymphoid tissue, which in our case, in the case of the eye, primarily are the draining lymph nodes in the cervical chain and the submandibular area, is where the T-cell mediated immunity is generated. So for the most part, when we talk of T-cell mediated immunity, whether it's transplant or infection, most forms of infection, or dry eye disease, as you'll see, we're talking about the interaction between APC and T cells in the lymphoid tissue, not in the eye. And then the blood vessels, as I mentioned earlier, are critical as a conduit for the delivery of either specific immune cells, antigen-specific immune cells, or cells such as those of the innate immune system that are not antigen-specific. And then these cells come out of the intravascular compartment, and so the more blood vessels you have, the more facile the delivery of those inflammatory cells to the cornea. Now, the relevance of this concept is emphasized here in this slide, where you can see in experiments done by a fellow in my lab just over a decade ago, here's a Kaplan-Meier survival curve of corneal transplants performed in mice. And about half of these transplants survive, half of them fail if they are left unmanipulated. However, if you remove the draining lymph nodes of the corneal transplant hosts, you get universal and indefinite graft survival. It's a classic example of immunologic ignorance, and it emphasizes a couple of important facts. One, that immunity is generated in the lymph node, and second, that there is a crosstalk between the eye and the lymphoid tissue in terms of the antigens that the eye is seeing. Nobody knew how this occurs because for a long time it was believed that the eye actually, the antigens are sequestered. The concept of antigenic sequestration was one of the principal dogmas of ocular immunology until even 10 years ago, because people thought you don't see inflammation in the lens, you don't see it in the cornea, XYZ, because the antigens are just kept in the eye and the immune system is blind to what is going on in the eye. In fact, that's not true. The cornea has antigen-presenting cells, and the cornea can get ingrowth of lymphatic cells. And also, and I won't talk too much about this, but we showed for the first time that the antigen-presenting cells, when they mature, when they upregulate their expression of MHC class II and other molecules called co-stimulatory molecules that are important for them to present antigen to T cells, when that occurs, they also upregulate a receptor called VEGFR3, a vascular endothelial growth factor, as well as other chemokine receptors, which permits them to interact and respond to ligands secreted by the lymphatic endothelium, as shown here in this confocal micrograph, to permit these cells in the periphery of the cornea to actually squeeze into these lymphatics and leave the eye. And when we first showed this work, nobody actually believed us until we did some pretty basic experiments now in retrospect, where we did these transplants, which were enhanced GFP transplants, so that the grafted cells can be seen as green, and then we could track the movement of these cells out of the eye. And in the bottom, you can see here that you can actually localize and enumerate the number of cells coming from the corneal transplant that are draining into the lymph node. Massive numbers. And the more inflamed the host tissue, the more of these cells that you see. And we showed, however, that if you block VEGFR3, as we show in these panels here, in a dose-dependent fashion, you can suppress the migration of these cells of the draining lymph nodes and thereby promote the survival of these corneal allografts. So by inhibiting or suppressing this constant kinetic of information going from the eye to the lymphoid tissue, you can significantly alter the induction or expression of antigen-specific immunity to corneal antigens. But as I mentioned, the cornea has this unique feature of being avascular and alymphatic. What are the factors that regulate that? So being a clinician scientist can sometimes be helpful. It's usually not, but sometimes it can be helpful. And we made this observation that when we see these patients in our clinics who develop non-healing epithelial defects, almost in a matter of 1 or 2 weeks, that is typically followed with ingrowth of blood vessels. And conversely, when the epithelium heals, whether it's with anti-inflammatory treatment or a bandage contact lens or other maneuvers that we might employ, when the epithelium heals, there is a regression of the blood vessels. So we made the simple hypothesis that there is something that the corneal epithelium possesses, the phenotype of the corneal epithelium, that can suppress the ingrowth of blood vessels or raises the threshold for ingrowth of blood vessels in the cornea. And we did these experiments. Here's a normal cornea. Here's the limbal vasculature, again shown here in green. If you apply an inflammatory insult to the cornea, you get this transient ingrowth of blood vessels sprouting from the limbus, but then they regress. If you remove the epithelium, you see a very similar response to the inflamed cornea. However, if you remove the epithelium and you then induce inflammation in the cornea, you get a robust ingrowth of blood vessels, which is sustained. We did a number of other experiments, and we identified this factor, interestingly, to be our friend VEGFR3. This is, interestingly, ectopic or epithelial, not endothelial or leukocyte, but epithelial expression of VEGFR3 selectively by the corneal but not conjunctival epithelium. This was confirmed at the level of the mRNA and protein. And the concept here is that if you have epithelial cells in the cornea that express very high levels constitutively of a receptor that can play an important role in angiogenesis and lymphangiogenesis, it can serve as a trap. So it's basically a classic example of a trap system, so that the corneal epithelium can express selectively high levels of VEGFR3, thereby binding bioavailable VEGF-C and D, which would otherwise bind VEGFR2 on the endothelial cells, induce angiogenesis, or VEGFR3 on the lymphatic endothelial cells and induce lymphangiogenesis. So this is very interesting. At the very same time, the Ambari group showed that the corneal epithelium can also have secreted FLT1 or VEGFR1 that also can act as a secreted trap system to bind bioavailable ligand and suppress angiogenesis. So the cornea is really a special tissue in terms of active regulation and maintenance of angiogenic privilege by expressing specific factors that raise the threshold for angiogenesis. It's really interesting, again, because here you have one molecule, VEGFR3, depending on whether it's expressed by the antigen-presenting cells or the lymphatic endothelial cells, that will promote antigen crosstalk between the cornea and the lymphoid tissue, and then, if expressed ectopically by the epithelium, can raise the threshold for angiogenesis by binding bioavailable ligand and preventing those ligands from binding their natural receptors on the vascular endothelium. So, however, we obviously know clinically that this system, like any other system, is not perfect. If the balance is perturbed significantly, for example, by very significant acute or sustained inflammation, you can get angiogenesis. Problem is, and I won't really get into this given the audience, but the treatments for corneal angiogenesis are far from perfect. They either don't work, or they're very expensive, or they require repeated treatments. So we've been very interested in coming up with strategies, molecular and cellular strategies, that could suppress corneal angiogenesis in a safe fashion. A lot of this work has been done here. I've listed some of the references down below. We've been looking at ways of targeting many different pathways involved in angiogenesis. Some are quite interesting and novel, such as our work in lipoxins and pro-resolution molecules like the resolvins, which are very, very potent anti-angiogenic molecules that, as we've shown, for the first time. But to go into the clinic, we began by going down the biologics path by looking at bevacizumab, otherwise known as Avastin, its trade name, and ranibizumab, otherwise known as Lucentis, which is the Fab fragment of the humanized monoclonal bevacizumab or Avastin. So one is about 1/3 the molecular weight of the other. So we began these clinical trials. We've had a number of INDs approved through the FDA to permit us to do this work. This was the first patient that we had several years ago, a 65-year-old woman with a history of herpes zoster keratitis, patient of one of my colleagues, Debbie Langston, again with blood vessels growing into her cornea. And as I mentioned, the blood vessels were amplifying immunity. She was having corneal stromal keratitis, breakdown of the epithelium. She required chronic on-off corticosteroids, causing ocular hypertension and glaucoma. Very, very typical, unfortunate picture that we're all very much familiar with, those of us who work in the clinic. So we enrolled her into our study. and we gave her anti-VEGF molecules topically, and within a week, we went from this picture that she'd had for years to this picture here on the right. And what is very interesting, and we have seen this again and again, and this is again because the thermostat level, if you will, for angiogenesis is really high, the threshold is set high. If these patients respond to anti-angiogenesis intervention, they respond quickly and they respond in a very sustained fashion. So the anti-VEGF was given to them for only 3 weeks. Here's baseline, day 7, week 6, 3 weeks after termination of the therapy. And here is week 12, 3 months afterwards, and still there is no sign of the blood vessel ingrowth. Again, something that required years of corticosteroid therapy. We went on to look at different parameters of blood vessel ingrowth into the cornea. We developed a computer quantitative system to look at vascular caliber and the neovascular area, and we showed that you can get about a 50% reduction with just a 3-week treatment regimen with anti-VEGF, in this case, Avastin. We've looked at comparisons with topical ranibizumab or Lucentis. That's shown in red as opposed to bevacizumab, which is a full-length antibody. And you can basically see a more rapid response to Lucentis. You see a response at 3 weeks that you normally would see about 4 months down the line with Avastin. However, the difference over the long term is not statistically significant between these groups. And we have a paper that just came out this month looking at the comparison between Avastin and the Lucentis in the cornea. Again, very similar looking at vascular caliber. Now, I just wanted to show this to you because those of you who are interested in immune modulation or modulation of really anything topically, you might be interested in protein-based or biologic approaches. And the question always comes up as to what is the degree of penetration of a biologic molecule, which is tens of thousands of kilodaltons, into the corneal epithelium. And the answer, we have a paper that I think came out in IOVS recently looking at this. And here you can see Avastin 1 hour after application of 1 drop. Here you can see Avastin 4 hours, and you can see that it's barely visible. So the normal cornea that has the normal epithelial barrier function intact is very resistant to biologic therapies actually getting significantly penetrated through the corneal epithelium. However, if the cornea is inflamed and thus the epithelial barrier function not intact, you can get very significant deposition of the protein of choice into the corneal stroma and even deeper, occasionally even beyond the cornea. And if those of you interested in this work should take a look at this paper in IOVS. We've been looking at other strategies and a new investigational new drug permit that we got from the FDA has permitted us to look at another factor, PDGF. This was based on work where corneal neovascularization was measured, and then either an anti-VEGF aptamer or an anti-PDGF was delivered, or a combination. And it was shown that a combination approach is more effective than a pure anti-VEGF or anti-PDGF strategy. And based on that, we just completed a trial in our clinic. I don't have the analysis of all the data to show you yet. We expect that to come out in the next couple of weeks. But here is one patient who received this treatment. It's called pazopanib. It's an inhibitor of VEGF and PDGF. Here's the baseline angiogenesis, and here's after initiation of therapy on your right. So in conclusion, here's one example of taking a strategy which has a good biologic rationale from the laboratory to the clinic, looking at development of topical biologics for application to the cornea. We're looking forward to beginning a multi-center trial this fall. It's a randomized double-masked trial looking at the use of bevacizumab. Intraoperatively, again, it's a controlled masked randomized trial. Intraoperatively and then topically post-op for 4 weeks in high-risk corneal transplantation to see if that strategy can promote graft success based on the paradigm that I showed you earlier, the role of blood vessels in delivering immune effectors to the ocular surface. We need to be careful when we talk translation because it's never as simple as we think. And we are very much aware of the fact that VEGF can be an important trophic factor for nerves and neuronal regeneration, especially in inflammation. And therefore, we've been very careful to utilize a very low concentration of our anti-VEGF and to also employ them for a short time, lest there be a toxic or a neurotrophic effect on the cornea. And this is work done by Mark Rosenblatt, who was one of the trainees that we had in Boston. He's currently at Cornell in New York. So I wanted to change gears now and go from, if you will, an example of a full-minute, very devastating injury, insult to the cornea that leads to scarring such as angiogenesis, to a more subtle and more chronic form of inflammation, which at the same time is the most common ophthalmological condition for which people seek attention in the Western world, in the developed world, known as dry eye disease. Dry eye is extremely high in prevalence. About 1 in 7 people in the US have some degree of dry eye symptoms. So there are millions of people who have this. And it's really an area of significant unmet medical need. Currently, there is one treatment that has been approved. It doesn't really work, as the clinicians in the audience know. In most people, it does work well. In a minority of patients, and still, despite that, the drug has a very large market, again, as a reflection of that unmet medical need. So we've been very interested in trying to understand what the immune mechanisms of dry eye disease are. Dry eye disease 10, 15 years ago was really thought to be almost entirely a biophysical problem. You know, you don't have enough water, the lids are rubbing on the epithelium, that causes discomfort, period. You know, it was basically seen simply as an issue of inadequate lubrication. And there was very, very minimal understanding of the role of immunity and inflammation in this condition. So we set out to investigate what some of these mechanisms might be. In particular, T-cell mediated mechanisms. To help us with this, we developed what we call the Controlled Environment Chamber, or the CEC. These are cabinets that we buy. They're quite expensive. They're developed basically for storage of microchips, and the military has a lot of this stuff as well. So once you know that's the case, it doesn't come cheap. But each of these, I think, costs about $5,000 or $10,000. And we have modified it to permit us to pump air, desiccated air, into the chamber. And then we use this system in order to dial the exact humidity level that we want our mice to reside in. And these mice, even without any anticholinergic agents, can develop significant dry eye disease within a matter of days. If you give them anticholinergic, which will aid in suppression of the lacrimal compensatory mechanisms in response to the desiccating stress, then you even will exacerbate the dryness. When you put them in the chamber, they have reduced tear secretion, and then we use the same clinical grading scheme that the National Eye Institute has recommended for clinical trials to assess the level of dry eye disease in these mice. They also lose goblet cells, which are mucin-secreting gel-forming mucin-secreting cells. So what do we know about the pathophysiology of dry eye disease? Well, very similar to other forms of even acute, whether they're infectious or transplant-mediated insult to the cornea, the first cells that respond obviously are the resident cells of the cornea. These are the epithelial cells, to some extent the fibroblasts, and the resident monocytic cells, or the resident macrophage type cells in the cornea. There's a very significant overexpression, and we've shown this both at the level of mRNA and protein, of pro-inflammatory factors such as IL-1, both alpha and beta, as well as TNF-alpha. So that's the first thing that occurs, and it can be measured in the cornea of these animals. As a result of this perturbation in the cytokine microenvironment of the cornea, what happens next? Here's the normal cornea. These top panels and the bottom panels here show the dry eye cornea. You get this gradual but significant recruitment of these bone marrow-derived cells of the cornea. The immunopathology of dry eye disease, unlike many other inflammatory phenomena in the cornea, is really characterized almost exclusively by the recruitment, mobilization, and activation of these antigen-presenting cells, as opposed to large numbers of T cells. And I'm not going to get into that, but as an aside, it's very interesting because we showed a year or two ago that the corneal epithelium, in addition to expressing the VEGFR3 trap molecule, also expresses very high levels constitutively of PD-L1. And this PD-L1 mechanism, programmed death ligand 1, can actively suppress the the infiltration of T cells into the cornea. Interestingly, that is also tightly regulated and is perturbed. And we showed in a paper that was published, I think, a year ago, that PD-L1 also regulates the expression of chemokines by the corneal epithelium. So the bottom line is, in dry eye disease, the immunopathology, unlike that of the conjunctiva in dry eye, where you get tons of T cells, is characterized by mobilization of these antigen-presenting cells. But very importantly, it's not just an issue of numbers. They also change phenotype. They acquire class 2 expression and co-stimulatory molecules, which will permit them to present putative autoantigens to T cells. I mentioned to you all at the beginning of the talk that, as I showed you earlier, lymphangiogenesis, as far as we know, always accompanies inflammatory angiogenesis, but the reverse is not true. And interestingly, this is the only situation where we've been able to show that there is a selective ingrowth of lymphatic vessels into the cornea without concurrent blood vessels. So we'll talk more about that a little later in the talk and what the molecular mechanisms of that might be. But here again, you see our yellow lymphatic vessels sprouting out of the corneal limbus, corneoscleral limbus, into the cornea in dry eye disease. Now again, what is the role of these lymphatic vessels? The role of them is to facilitate the migration of antigens and antigen-bearing, antigen-presenting cells from the cornea to the lymphoid tissue. And these flow cytometric studies here on the right show you that if you look, for example, here at CD11B, class 2 positive, 11B positive cells in the draining lymph node, a classic marker for activated or mature antigen-presenting cells, you can see that there is a very well-defined population after the development of dry eye disease that you don't have before the dry eye is induced. So again, here's a link between lymphatics and their possible role in generating immunity. So again, I wanted to emphasize this, that dry eye is a very nice model without concomitant blood vessels or many of the other confounding variables that you might see in acute forms of insult, such as chemical burn or angiogenesis, for APC activation, mobilization, and then selective ingrowth of lymphatics. So it's a very nice model, actually, to study corneal immunology, beside the relevance of dry eye disease. Now, in summary, what do we know about the T-cell-mediated aspects of dry eye disease? A lot has been going on both in our laboratory and the laboratory of Steve Fluchfelder down in Baylor. Very, very, frankly, interesting and reassuring to know that despite totally independent laboratories, we have had very similar findings in many respects. So we believe, and similarly to work done in Steve's laboratory, we have implicated the IL-17 secreting T helper cells, the CD4 positive, CD4 positive IL-17 secreting cells, the Th17 cells, and the interferon gamma secreting Th1 cells as being the main effector cells that mediate disease. So again, putting it back in our paradigm, we have shown, and again, I'm sort of going through this quickly, that the principal T cells that are activated in response to these antigen-presenting cells migrating in dry eye disease are these two T cell phenotypes. Now, the role of IL-17 in autoimmunity has been getting a lot of attention, and this is again a classic examples of an autoimmune form of ocular disease. Levels of IL-17 are significantly overexpressed, both in the lymphoid tissues and on the ocular surface, if you compare normals to dry eye. And this is again work done in both mice and animals. Pardon me, mice and humans. The question, though, is, after we got to this point about 2 or 3 years ago, everyone said, okay, fine. So you have the T cells and you have the inflammation. Is this simply a consequence of the dryness? So that if you treat, for example, the dryness with lubrication, or you just let the lacrimal gland compensate by secreting more water, the inflammation will go away? Or do these, up to now, phenomena really mediate or induce disease and maintain disease? So we did a series of experiments that we haven't published. The manuscript is just about to be submitted, which I think are quite telling. So what we did is this. We induced dry eye disease over a 2-week time course. You can see this on the x-axis in days. So we begin here and we go for 4 months. So we have 2 y-axes here. On the left, we're looking at corneal fluorescein staining. The higher the score, the worse the dry eye. On the right, you have tear production, shown here in purple. And what you can see is that as the corneal fluorescein staining goes up, as the dry eye is induced, tear production goes down. What we do then is we take out these mice out of the chamber, we put them out in the room air, and we let them live there happily for months. And what you can see is that, not surprisingly, the tear production goes actually to supranormal levels. Here's the normal level. It goes to supranormal level. And despite this, after many months, there is still chronic epithelial disease which never normalizes, even 4 months after we initially induced dry eye disease. So there clearly is a chronic form of dry eye disease that is maintained. Now, what maintains that? So again, by acute dry eye, we mean the 2 weeks. Chronic, it means outside of the chamber, no anticholinergic therapy, nothing, if you will, just left alone. Now, if you look at the cytokine signature in acute, the normal is shown in white, the acute in gray, and the chronic 4-month dry eye in black. You can clearly see that there seems to be a more interferon gamma-mediated disease at the early on, and more of a shift toward IL-17 in the later stages of the disease. And to characterize this further, what we did was that we collected cells and stored it for the CD4 positive T cells from normal mice, the acute dry eye mice, and the 4-month chronic dry eye mice. And then we adoptively transferred them to normal mice, and we put them into the chamber without any scopolamine treatments, as a challenge, if you will, to see whether these mice will then show any sign of what would otherwise be called a secondary response, if you will. And what we saw was that we could induce much more severe dry eye disease, and it happens more quickly, quite characteristic of a secondary response, in animals that were adoptively transferred with cells that were harvested from the chronic dry eye mice than those that had had the acute dry eye disease, despite the fact, if you recall, that the level of staining and dry eye severity was worse in the acute setting than the chronic setting. So the acute is the light blue, the purple is the chronic dry eye, and here are those getting the normal non-dry eye T cells adoptively transferred. And then if you look at the expression of interferon gamma and IL-17 in the draining lymph node in these adoptively transferred animals, you can see a trend which is very comparable to what we saw in those initial animals that had not been adoptively transferred. Again, here we're looking at the normal animals, the dry eye disease transferred with the normal T cells, the acute dry eye shown here in the gray, and in the black showing the chronic dry eye. And you can see again the very significant elevation in IL-17 by the T cells in the animals that had received the chronic dry eye disease CD4 T cells. Now, we've done a lot more characterization of these cells, and these cells you can clearly see that there is a distinct population of CD44 high, CD62 ligand negative classic memory T cells that are generated in these chronic dry eye disease animals. And in subsequent adoptive transfer experiments, we have stored it specifically for these distinct populations, and we can recapitulate the most severe disease by adoptively transferring these IL-17 secreting CD44 high, CD62 ligand negative cells, generating the worst kind of dry eye, and again, demonstrating classic T-cell mediated disease. So dry eye is a chronic T-cell mediated disease, and continued desiccating stress or hyperosmolarity is not a requirement for maintenance of the disease state, as we have shown. And the chronic aspect of the disease is basically characterized by enhanced IL-17 or overexpression mediated by these memory T cells. Wanted to switch some gears and go back to transplant in the setting of dry eye disease, because it's a nice model to study T cell regulation. Now, as I mentioned earlier, dry eye is very common, and therefore transplanting corneas in dry eye hosts is also very common. But the clinical dogma is, which we believe is true, that the corneal transplants don't fare as well in dry eye disease patients, but for reasons that are, again, not known. People have been thinking, well, it might be that the epithelium is perturbed and there's just chronic inflammation, et cetera, et cetera. So we wanted to look into this further. This is, again, all unpublished and work in progress. So again, we have our normal mice, our mice in the chamber who developed dry eye disease. And what we did was that we did a classic fully MHC mismatched graft from these B6 mice into BALB/C dry eye or normal mice. And if you look at the survival here, as I mentioned earlier, about 50 or thereabouts % survive if they are normal hosts. And if they are dry eye hosts, there is a significantly earlier and more higher incidence of rejection if the transplant is done in these animals. Now, why is that the case? Now, we had a paper a couple of years ago showing that acceptance of corneal allografts is associated with the function of high FOXP3 expressing regulatory T cells. What are regulatory T cells? Over 30 seconds. Regulatory T cells are also CD4 positive T cells which respond to IL-2. They respond to other cytokines. They are phenotypically different from other CD4 T cells by their character of expressing very high levels of TGF-beta and IL-10, which are immunoregulatory or immunosuppressive cytokines. So the secretion of immunoregulatory cytokines by these T regulatory cells can suppress the effector T cells. Also, these T cells, these regulatory T cells, express co-inhibitory factors like CTLA-4 that can impact the functioning of antigen-presenting cells and making them less sensitizing in their presentation of antigen to naive T cells. So again, we showed that animals that reject their corneal allografts have demonstrated dysfunction in their T regulatory functionality at the level of their lymphoid tissues. So we were interested to look at the functioning of T regulatory cells in the setting of dry eye disease in transplantation versus in the non-dry eye environment. First of all, we showed a couple of years ago that the level of FOXP3 expression in of itself can be utilized as the most proximal determinant of their capacity to suppress proliferation. So we looked at the levels of FOXP3 expression, looking first at MFI at the level of the protein. And interestingly, you can see that in both cases, both in dry eye and in room air, the level of FOXP3 expression is less in the rejecting mice than in the acceptor mice. But interestingly, even the accepting hosts have lower levels of FOXP3 expression than the accepting mice that are outside in normal room air. And this is despite very minimal differences in terms of the overall numbers of the Tregs. These Tregs are defined as CD4 positive, CD25 positive, FOXP3 positive. Very, very similar trend if you look at mRNA levels, here done with real-time PCR, again showing lower levels with rejectors than acceptors and lower in dry eye than non-dry eye. Very similar if you look at their function. So if you do a proliferation assay and you look at their Treg suppressive capacity, you see that there is this defect, probably due to lower expression of FOXP3 by the Tregs in the dry eye hosts. How is that defect induced at the molecular level? We're in the midst of those experiments looking at co-inhibitory factors and how those might be suppressed in these Tregs. So we did the following experiment. We said, can we reconstitute normal Treg function in animals that are kept in the dry eye environment? So we got a naive biopsy, and we adoptively transferred either these natural Tregs or non-Tregs that are CD25 negative. We did this 24 hours prior to transplantation, and then we gave them the allografts from the B6, normal B6 mice. And interestingly, we reconstituted these mice with normal Tregs. So if they got normal non-dry eye Tregs, but they were dry eye hosts, we actually showed that the rate of graft rejection is very comparable to a normal room air control hosts, and certainly much, much different than those that got the adoptive transfer of the non-Tregs. So in conclusion, dry eye disease obviously affects corneal transplant survival, and there is a defect in the expression of FOXP3 and a defect in the functioning of the Tregs in dry eye disease. This leads us to the obvious. So strategies that would either promote homing of Tregs, and we have done a lot of work in that area, which I won't be able to share with you due to time, or ways of ex vivo expanding Tregs and then transferring those to the host would be potential therapeutic strategies for addressing the Treg defect in dry eye disease. The last thing that I wanted to show you in the last couple of minutes was to go back to factors that regulate lymphangiogenesis in the cornea. And again, I wanted to take you back to this concept in dry eye that lymphatics are critical for permitting trafficking of antigens to the lymphoid tissue and then the blood vessels in the homing of these cells. So we became very interested in the regulation of lymphatics on the ocular surface. And here's data that we published a couple of last year looking at anti-lymphangiogenic strategies such as anti-VEGF-C. Here's a score of the severity of dry eye disease, and you can see that you can suppress the severity of lymphatics and thereby suppress the severity of dry eye disease. Now, is there a link between IL-17 and lymphatic growth? And the answer is yes. So we placed pellets in the mouse cornea containing either IL-1 beta, IL-17, or IL-17 concomitant with IL-1 beta suppression. And again, if you can see here, IL-1 beta pellet leads to tremendous ingrowth of blood vessels, shown here in green. IL-17, less blood vessels, significant lymphatics, but if you block IL-1 beta while you have the IL-17 pellet in the cornea, you suppress significantly the blood vessels, but you don't suppress the lymphatic vessels. And this correlated very, very nicely with the selective overexpression of pro-lymphatic VEGF-C and VEGF-D. So again, here in a co-culture system, we show that corneal epithelial cells that express IL-17 receptor, when stimulated with IL-17, can secrete very high levels of VEGF-D, which is a ligand that specifically and exclusively binds VEGFR3 that drives lymphangiogenesis. Again, showing a link between what we believe is a Th17 product, maybe now a PMN17 product, and lymphangiogenesis in the cornea. So IL-17 clearly has a pro-lymphangiogenic function. It promotes development of lymphatics, and thereby it promotes the trafficking of antigen and APC into the cornea. And we've looked at utilizing anti-IL-17 antibodies for suppressing dry eye disease. We've done this both systemically and topically. And here's an example here showing you a whole mount, confocal whole mount of the cornea. Again, the orange shows the the lymphatic vessels, untreated. And if you give anti-IL-17 topically, you get a tremendous suppression of the lymphatic vessels. And as a result of that, you see a significant suppression in the severity of the dry eye disease, either if you give it at the beginning or after you develop dry eye disease. If you suppress IL-17, it can significantly block the severity. So in closing, this is sort of how we see the immunopathogenesis. We see the activation of antigen-presenting cells. We see a role for IL-17 in promoting the trafficking of APC to the lymphoid tissues. Both Th1 and Th17 subsets are primed and expanded. And then the Th17 cells also seem to be particularly resistant to Treg suppression. And then the homing of these effector cells through the expression of specific chemokines on the ocular surface, in particular CCR6 ligands, as we showed at the recent ARVO. And then, as I mentioned very briefly, the constitutive role of PD-L1 by the epithelium in suppressing the infiltration of the T cells into the cornea. They get in the conjunctiva, secrete cytokine, and cause disease. So in closing, dry eye disease is T-cell mediated, and IL-17 plays a critical role in that process. I wanted to thank the principal individuals in the laboratory that have contributed significantly to this work, and the funding mechanisms that have made the work available. Thank you very much for your attention.
Questions? So the dry eye, you talked about the Th17 cells, but you didn't talk about the Tregs. Are they in relatively low numbers? Or have you looked at ratios of Th17 versus Tregs later on in the dry eye disease?
Yeah, the Treg numbers, we don't see very significant changes in Treg numbers. The real change that we see is the level of the FOXP3 expression. Yeah.
So in the...
So FOXP3 goes down. We don't, again, it's not definitive. CTLA-4 IG goes down, co-inhibition goes down, IL-10 goes down. So I didn't show you the IL-10 data. Very similar, very comparable to the functional proliferation assays and the FOXP3 levels. So the function of these Tregs is clearly lower in dry eye disease.
Do you see that if you're taking an approach of treating of targeting IL-17, then that should have an effect both on reducing Th17 cell activation and blocking angiogenesis at the same time?
Yes. And countering the selective resistance of the Th17 cells to Treg suppression, which I didn't get into, but that we published that in JI a couple of years ago, about 2 years ago. So the Th1 cells, despite the Treg defect, the T regulatory cells can suppress the Th1 cells, but not the Th17 cells. Yeah.
How does cyclosporin or specifically Restasis, have you tried that in your mouse transplant model?
It doesn't do anything in transplant. In dry eye disease, it has a very modest effect. Yes. It's not a very, it's not a big surprise in that nobody really knows where topical cyclosporin works. And if you think of the mechanism, it primarily hits the IL-2 mediated mechanisms. So much of the IL-2 is really functional at the level of the lymphoid tissue. So if you could get the cyclosporin to the lymphoid tissue, you would expect it to be more effective than on the ocular surface. Of course, right now, there's all this interest in IL-2, low-dose IL-2 in expansion of Tregs, right? IL-2 has a tremendous effect on CD25 expression. So you really can expand Tregs by giving IL-2, and people are doing that in a whole host of autoimmune diseases like rheumatoid arthritis and GVHD now. Yes?
What strategy you're thinking about for converting these rogue T-cell corneal mice that you've dried up for a couple weeks and converting them back to normal?
Converting the mice or the T cells?
The mice. I mean, is there a way that you can convert this chronic disease back to normal?
I think it's a tall order because of the involvement of the memory T cells. I think theoretically, if you could administer an IL-17 antagonism over some period, you would expect these memory T cells to eventually die off and not be relevant. I guess that would be the only strategy I can think of.
Transfer Tregs specifically? Would that do it?
Functional Tregs to those to those chronically? No. Similar to what we did for the transplant. Well, I mean, these are tough experiments because they're so long. One of the beauties of corneal research is, pardon me?
Tregs last for?
No, sure. I'm just saying that they're lengthy experiments. So, yeah.
Yeah, it's 4 times 1, 6 months.
Yeah.
Then I have a question on angiogenesis.
Yes.
The topical application of the antibody to VEGF resolved the angiogenesis very effectively in a short period of time.
Yes.
What happened to the stimulus, the angiogenesis stimulus, whatever was causing the angiogenesis? Was it still there after you treated antibody?
Yeah, that's a great question, and that really goes back to the issue of the stimulus inducing angiogenesis and angiogenesis stimulating the capacity or amplifying the capacity for that stimulus to promote angiogenesis. And so for the case of, let's say that zoster patient whom I showed you with the chronic angiogenesis being there with the episodic stromal keratitis, those blood vessels were there simply because the episodic stromal keratitis was adequate for the maintenance of those active blood vessels. And the way that I see it is that unlike, for example, choroidal angiogenesis, which requires repeated anti-VEGF, in cornea, because you have anti-angiogenic mechanisms, you have the TIMPs and you have the soluble VEGFs and you have the membrane VEGFR3, et cetera, the thermostat is just set at a different place. So it's enough for you to stop it for some time and then you don't see the recurrence of that angiogenesis. And we see that repeatedly. And if you noted that in almost all of those graphs, despite the fact that the treatment was for 3 weeks only, even for months you would see that decrease in the degree of angiogenesis. So if they respond for the long term. You don't have to go back in and give it again. And again, I think obviously it's a balance. All of these patients had stable angiogenesis. I failed to mention that, so we did not treat patients with acute angiogenesis where you would expect the angiogenesis to regress normally as the infection clears or the insult clears. So it's a very nice contrast to the choroidal NV or PDR where you have to keep going back and giving additional treatments. Thank you.